Yeah, so VAL-1221 is uniquely designed that has an antibody GA fusion. So the antibody portion is designed to deliver more of the GA enzyme to the tissues that's needed, muscle, skeletal muscle, heart, but also drive intracellular penetration. GA alone can penetrate cells through mannose-6-phosphate receptors to the lysosome, but the addition of the antibody portion can also enter cells to the cytoplasm. And interestingly, in Pompe disease, the GA is deficient in the lysosome, but in often cases you get glycogen that spills out of the lysosome into the cytosol or autophagic vacuoles that are extra-lysosomal. So this is designed to be able to clear lysosomal glycogen, but also address that extra-lysosomal glycogen that could be present in Pompe disease patients. Trial was a phase 1/2 clinical trial designed primarily for safety and tolerability, and that has done very well in that regard. I think it's safe to say that it seems to have a safety profile similar of the GA alone. We have completed an extension upon that study, and some patients have now been treated up to 18 months. The study is now closed. Unfortunately, my previous company, Valerian Therapeutics, ran into funding problems. And so I've actually founded a new entity called Parasail, where we've transferred all the previous assets, so the VAL-1221 intellectual property, the drug material, into a new entity. So I'm currently in the process of raising new funds that I ideally would be allowed to, a new study to be developed, both in Pompe disease, and we're also looking to expand into other polyglucosan disorders and glycogen storage diseases, because really the unique property that I mentioned for 1221 is that we could clear also extra-lysosomal glycogen. And in most glycogen storage diseases, that's the case, that glycogen buildup is actually cytoplasmic. And so one case we're looking at is, could we expand even beyond Pompe to these other areas? And in Pompe, we may want to tailor a study that could look at patients that a little earlier and more severe cases, because in this current ongoing study that was completed, it was adult onset patients that had been on previous treatment, myozyme or standard of care, for over a year. And those patients typically didn't present with high glycogen buildup in the muscle. Actually, they had, in many cases, probably lost a lot of fast twitch muscle fibers. And so glycogen's not elevated. In the case of our drug, it really needs an elevated glycogen to have its function, because it can only clear elevated glycogen. So if it's not elevated in specific patients, you may miss out on the potential benefit, the unique benefit of this drug. And so I think in a new trial, we'd really want to look at muscle biopsies and try to identify patients that definitely have not only elevated glycogen, but some potential cytoplasmic or extra lysosomal glycogen.